I want to thank the organizers for inviting me, and I am a co-organizer. So then I want to turn it on you and thank you very much for coming today. It's a really great gathering, and we've already discussed some incredibly important concepts and not just data. So I want to talk to you today about the many menopauses, because there's diversity in people, but there's also diversity in conditions. And menopause has been thought to be a monolithic kind of thing that all women go through. And I want to disabuse you of that notion and tell you a little bit about how different menopauses can affect women's brains and their aging trajectories differently. But first, I forgot to say that I'm Gillian Einstein. I'm the Wilfred and Joyce Postlins Chair in Women's Brain Health and Aging, and I'm a professor at the University of Toronto. So I don't have any conflicts of interest, unfortunately. And my plan today, again, is to talk about menopause as a much studied, that inflection point that Rachel just described, a much important turning point for women's cognition on the road to dementia, but really focus on the many menopauses, and one in particular, surgical menopause. Describe a little bit of the data that we have in my lab, just a smidgen, and then tell you about how women experience these decrements, and a little sum. So the North American Menopause Society, as well as the Endocrine Society, published guidelines on menopause many, many years ago now, describing 4 types. There's spontaneous menopause, which occurs roughly, somewhat after the age of 50. I think 51.8 years was the number given earlier. An early menopause, which is between the ages of 40 and 45 years, which is the absence of menses with no clinical reason. Premature menopause, which is less than 40 years of age, which could include premature ovarian involution, which is a kind of waxing and waning of ovarian function. And then induced menopause, which is also usually before the age of 49. It's can be caused by chemotherapy. It can be caused by hysterectomy, which leads to ovarian dysfunction. And it's not that removal of the uterus by itself leads to menopause, but if you damage the blood supply to the ovaries when you're doing the surgery, you can actually get ovarian failure. The actual removal of the ovaries, oophorectomy, which it's kind of the one I'm going to talk about today, is called bilateral salpingo oophorectomy, where you remove the fallopian tubes as well as the ovaries, and then ovarian ablation via radiation. And each of these actually has distinct hormonal changes leading up to and after menses cessation, and each has a unique health and cognitive consequence. So many years ago, we wrote a little paper called The Many Menopauses, published in, I guess it was Menopause, the journal Menopause. And we described a bunch of papers that we reviewed between, I don't know, maybe it was between 2012 and 2014. It'll come up here on the screen at some point. And we basically described these papers as either undifferentiated with no information at all on menopause or time since menopause in their groups. We came up with something called demographic differentiation, which had some descriptive information, either reporting mean age at menopause, time since menopause, or percentage of participants from each menopausal type. But these factors were not considered in the analysis. A partial differentiation, which provided many of these same points of information, but for example, the article might consider the age at menopause and use it in the analysis, but not the type of early menopause. So, it's complicated. I just got a critique of a paper that we submitted, but that we're working on for publication today, where the person reading it said, we can't tell the difference between what's BSO and what's age at menopause. Well, it's really hard to differentiate those. They're really not differentiable. And then there were a few, but a very few papers that did full differentiation, where they talked about either the different types of menopause that are in their studies and included that in the analysis, or they only studied one menopausal type, which actually is the strategy that we've taken in my lab. So it just shows you, here we go. The literature in 1 year, 2016, which was post these recommendations. And you can see that I wonder if I can get the pointer.
Yep, it's on the.
Is it on the screen?
Yeah, if you look back, it's.
Okay. Oh, here it is. That group right there is the undifferentiated. So most of the studies were undifferentiated. Gosh, it's hard for me to see this. It was the same problem in both observational studies and clinical trials. So observational studies are on your left and clinical trials are on your right. So I'm not going to go into the details. You've just seen everything, and I don't want to go over time. But the important point here is we need to consider the type and age of each kind of menopause in order to allow for more rigorous cognitive research. And I would postulate that maybe some of the problems in the mixed literature around different types of hormone replacement is not just that they're using many different types of hormone replacement, but they're throwing in many different types of menopause in there as well. The menopause that I want to talk about today is induced menopause, and the repercussions are immediate. Induced menopause by ovarian removals correlated with decreased verbal memory function if estradiol isn't replaced within 3 months post-oophorectomy. Exogenous estradiol improves verbal word recall after 2 months. It's correlated, but if not, it's correlated with decreased verbal memory. Age of oophorectomy is correlated with increased all causes of death and dementia risk. And here, I think this is pretty interesting in the context of risk and resilience. So Barbara Sherwin's studies showed significant but small differences, as do our studies. But this is an Egyptian study where the women had an average of 2 years of education. And you probably can't, I don't know if you can, I guess you can see it, that the effects are incredibly strong at 6 months post-oophorectomy in every category: digit span, mental control, logical memory, associative learning, visual reproduction. It's not so, it's not so strong at 3 months, but at 6 months, it's much stronger in some ways than many of the things that we see in our study in a highly educated cohort. So I think this really speaks to the importance of education for any type of menopause. And then the earlier the induction of menopause leads to a steeper slope of cognitive decline. And in Riley Bove's study, she found an increase in global measures of the burden of AD as well. But you can see in this graph that the sort of blue-purple on the bottom, that is the oophorectomy group that were younger than 38 at the age of oophorectomy. So the younger the age of oophorectomy, the worse your cognitive outcome is. And not only that, but you have a higher burden of plaques and a higher burden of tangles. So she was lucky enough to get the brains on autopsy and was able to show this. And from the Mayo group, Kajal Kantarshi has shown that if you image the brains of women with BSO prior to the age of 46, but at the age of 63, you're imaging them, you see smaller amygdala, thinner parahippocampal, entorhinal cortices, and fractional anisotropy is lower in the entorhinal cortical white matter. There's no difference in PIV score, no difference in cognition, and no effect of hormone therapy. But most of the women in this study, almost all of them were on hormone therapy, and all of them were on conjugated equine horse estrogens. And if you want to know why I think that might not be the greatest idea, we can talk about it later. Also, sleep after BSO is worse in women. There's worse sleep duration, worse sleep efficiency, and more insomnia. So there are a number of decrements that are conspiring to poorer cognition or greater risk factors that are building. In my study, we're studying women who carry the breast cancer mutation gene. Those women are recommended to have a prophylactic bilateral self-pingo oophorectomy as soon as they're done in childbearing. And I think this is a gendered condition because I don't think anybody would tell men to have their healthy testes removed just because there was a risk of testicular cancer. But these women are between a rock and a hard place because this happens to be the most effective therapy for them. There are no smart neurophysicist that's figured out a way to image the ovaries over time in order to detect new cancers, which gets to Rachel's point, why not? So it's in some sense very good for the women in terms of their survival rate from cancer. And it's not a small problem because in the US, there are over 300,000 bilateral self-pingo oophorectomies done a year. We don't have stats for Canada. So we're studying these women. We're doing an accelerated longitudinal design. I was so happy to hear that expression mentioned earlier. We recruit women from 1 to 10 years post-oophorectomy. Each woman is tested once a year for 3 years, and she provides information basically from 1 to 13 years post-oophorectomy. And what I'm going to present today are our results from spatial working and verbal episodic memory, some of the imaging. We also do sleep. We measure estradiol. We measure progesterone, we measure ApoE14 genotype, we look at cytokines, we look at other genetic markers and other aging markers, and I'm going to talk about the qualitative interviews and the body mapping story. So this is just an example of, some of our demographics. You can see that women are pretty well matched on age, they're pretty well matched on education, they're highly educated, BMI is not too bad. They're matched on age since BSO. I think the one thing they're not matched on actually is a history of cancer treatment. We only recruit people who are at least 6 months post-cancer treatment of any type. So we don't recruit anybody on aromatase inhibitors. And you'll see a little validation study that's tacked onto this. So we find that BSO reduces 17-beta estradiol and progesterone levels right away. And you can see here that women with BSO who are taking estradiol therapy have roughly matched levels of estradiol to the AMCs. And the ones who are not taking it have significantly lower estradiol levels. PDG P4 is all over the map because of the range of types of progesterone given and also the amounts. We find that this reduces verbal episodic memory. And you can see that the BSO groups are significantly lower than the controls, both for immediate and delayed recall. In our hands, estradiol therapy does not maintain episodic verbal memory. We do 2 spatial memory tasks. We do digits backwards and we do a spatial working memory task. And these have the most robust results. And what's interesting about these is, while it's interesting that there's a significant difference between the controls and the women with BSO who are not taking estradiol replacement. Estradiol replacement seems to help maintain this form of memory. So what I love about this is it shows that estradiol treatment is also very specific to memory domain, and we have to keep that in mind. When we looked at the dentate gyrus with Rosanna Olson doing manual segmentation, we found that in the CA2-3 region only, there was reduced volume between the women who had BSO and controls, and the women with BSO and estradiol therapy were doing as well as the controls. So here, estradiol therapy helps maintain volume. And Alana Brown, really with Cheryl Grady and others, found that there was a difference between when she included women in spontaneous menopause, which most people think of as natural menopause, but spontaneous menopause. She found that the real difference here was between age-matched controls and spontaneous menopause in the associate memory, novel versus repeat pairs encoding. And this is on the Sperling task. But when she looked in the brain, she found that the activation for novel versus repeat pairs encoding was similarly decremented in the BSO and the spontaneously menopausal women. And you may say to yourself, so, okay, so women with BSO are like spontaneously menopausal women. Yes, but the women with BSO are on the average 10 years younger than the women in spontaneous menopause. So this is maybe a premature aging effect that's important to keep in mind when we're talking about life trajectory. So when we asked the women and did qualitative interviews, are you having any problems remembering things or concentrating? One woman said, I started noticing memory problems. The issue is primarily the speed of recall. I can eventually remember the name that I'm searching for. It just seems to take far more time. I noticed that this lag with names or people, as well as names of books and places, it's as if I have a blank space opens up in my brain and I'm trying to fill it with all the information that I know should be there. And, these are highly educated women. They're very articulate about what the problem is. And we say, well, is this attention? And then post BSO, I started noticing I no longer remember conversations, people's names, logical memory. It took a lot of effort to recall where I'd place things, if not in their normal spot, spatial working memory. Lack of focus, concentration, particularly at work. So they're these are strong stories. And then we decided we would look at something called body mapping storytelling, which is a way of finding out more about their experience and what we would call embodied memory. And I won't tell you how we did this. It's on this on this slide, but we found that 5 themes emerged. And maybe the 2 I'm going to tell you about are chaos around the head and memory tools. So I was particularly interested in an idea about distributed cognition or distributed memory. Maybe they had memory other parts of their bodies and not just in their heads. So chaos around the head. The main thing that emerged is that there's chaos around their head. This is the term one woman used. You see these beams or pieces of paper streaming outside of this woman's head. This woman has a mass of sort of brain tissue that's all kind of mixed up here. And this woman portrayed an effusion of stuff coming out of her brain with lots of little bubbles circulating afterwards. And this was found in the other theme as well. But to tell you about that theme of memory tools, they all have memory tools. So this woman actually gave us her journal from the year that she was diagnosed with the BRCA mutation. She put it on the poster. This is how she kept track of things. She's also got to-do lists on here, but also shows bubbles coming out of her head when she tries to remember things. This woman has list after list after book after book, the purple, the green, the orange. These are different diaries that she keeps about different kinds of memories that she has. And I came to use the term memory hygiene because she's been doing this since she was like 18 years old. And it seems to help her a lot. But when I actually looked at the scores for this woman, her spatial working memory score was terrible, even though her verbal episodic score was pretty good. And then again, this woman lists of things, they exercise, they do a lot of self-care. This woman on your left actually has a little pile of raspberries or blueberries. She pays attention to eating. So there's a lot of self-care and a lot of attention to things that are supposed to improve memory. So I want to leave you with this message that it's important to acknowledge individual differences, that all menopauses are not created equal. To understand cognition specifically during the during menopause, we have to acknowledge what type of menopause we're looking at. Estradiol levels drop precipitously with oophorectomy, and there are decrements in tasks associated with early signs of AD. Their different menopauses have different cognitive, brain, and experiential effects. So I like to think of this in terms of something that I call situated neuroscience. I borrowed this term from a social studies of science expert named Donna Haraway, who talked about situated knowledges. And situated neuroscience is about putting our work in the context, thinking about early life events for later outcomes, taking into account the stages of life and the circumstances. How did they have their menopause? What was their life like during their menopause? Report individual responses to understand the details of variability. And, maybe most importantly, do research that matters to participants. And especially when we're talking about participants who have traditionally been excluded from research, that is everybody who's not weird, find out what the experience is like for them. And I think what we're coming to is more and more specificity about who is in our studies. And as I asked earlier, should generalizability actually be a goal, or take a notice from precision medicine and start to say, we're learning about individuals, what is important for this particular person. So I want to thank my students and collaborators and funders, especially the Women's Brain Health Initiative started by Lynn Coslins. And I'm just going to stop there and thank you.